Thanks to the hard work and dedication of scientists and physicians working under the guidelines and regulations of the Food and Drug Administration, we have many medications that allow us to treat previously incurable diseases such as HIV/AIDS, cancer, and hepatitis, diseases that previously would have been basically a death sentence for a patient with that diagnosis. Now we have the ability to allow these patients to live longer, healthier lives with a much improved quality of life. Whether we're talking about over-the-counter medications for things such as inflammation or pain, things we take for cold and flu, or prescription medications that we take under the guidance of our doctor and receive from a pharmacy, we now have the ability to treat many types of diseases that were not possible in the past. Despite the advances that modern medicine has made in providing us with these important medications, the Food and Drug Administration estimates that there are several thousand diseases that affect human beings. And of that several thousand, only about 500 have FDA-approved treatments for that disease. This leaves many, many diseases affecting humans with no current treatment. Complicating all of this is the fact that drug discovery is an incredibly time-consuming and expensive process. For this week's Ethical Dilemma in Science, we're going to take a look at the FDA Modernization Act 2.0 that was signed into law in December 2022 by President Biden, and we're going to consider, is it time to stop animal testing? The FDA Modernization Act 2.0 basically authorizes the Food and Drug Administration to consider data generated from alternatives to animal testing when examining the safety and effectiveness of drugs. Some of these non-animal alternatives could be things like cell-based assays that are occurring outside of the body in the laboratory, or things like artificial intelligence-linked computer models. The Food and Drug Administration was created in 1906 as part of the Pure Food and Drugs Act as a result of conditions at that time. In 1906, Upton Sinclair had just published The Jungle, which raised concerns about the really atrocious conditions in the meatpacking industry and stockyards. There were many cases of snake oil remedies in which the remedies were basically put together with no medicinal value and could cause problems. Therefore, the Food and Drug Administration was put into a place to ensure that there was a safe and effective and efficient supply of food and medications. Since 1938, the Food and Drug Administration required that potential drugs be safe. Since 1938, the Food and Drug Administration required that potential drugs be tested for safety and efficacy, how effective the drugs are going to be at working at treating that disease. The way that this was done was testing in animals, which include toxicity tests in at least one rodent species, usually a mouse or a rat, and at least one non-rodent species, something that could be like a dog or a rabbit or some other organism. So as illustrated on this slide, the drug discovery process, we can see that even before clinical trials start, the clinical trials are down here in green, we're looking at lots of chemicals, lots of compounds that are of potential interest that are examined to determine whether or not they could be used in human beings. But what we can see is that through that process of drug discovery and preclinical research, again, done on experimental settings, maybe on rodents, maybe on alternatives, but basically many of these compounds are shown to not be effective for the treatment of disease in human beings. And so they fail before they get even to the point of clinical trials. The opponents to animal testing have long argued that using animal models of human disease is not an appropriate way for testing whether something would be safe and effective in a human being. They argue that these Animal models are just simply that. They're going to mimic the human disease, but they're going to imitate it. They're not going to be the true disease in the human being. And therefore, a drug treatment that works in the animals may not necessarily be effective in treating a human disease. And so we take a look at this high failure rate of drugs in this development process, and we're going to see that it takes a long period of time, often 10 or 20 years from initial idea to finalization of the drug testing and ultimately FDA approval. It's becoming more and more expensive to develop these new drugs. It's estimated that it may cost up to $2.6 billion, billion with a B, to investigate fully one of these new possible medications. And that high cost and that high period of time to create that new drug could result in the fact that these pharmaceutical companies may only focus in on these most profitable drugs and leave many other important medications basically not investigated and remain without an effective treatment. On the other side of the argument are supporters for the continued use of animal testing. These supporters acknowledge the challenges associated with using an animal model, but argue we don't have a better alternative yet. They would argue that a number of Nobel Prizes in medicine have been awarded for use of animal models. We've learned important things, as illustrated on this slide. And these non-animal alternatives will be great, but we're not quite there yet. They're not fully understood. And so if we're looking at growing cells outside of the body in a laboratory setting, we may be able to mimic what is going on in a portion of the body, but we may not be able to show what's occurring in the entire body. If we take a look at how medications are processed within the body, many of us take our medications orally, and those medications would be broken down within the digestive system. And so we need to understand how those metabolites are going to be acting, how the breakdown chemicals from the drugs are going to be acting in our body. It'd be great to use a computer model, but the computer models are only going to mimic and illustrate what we are able to understand and program into the computer systems. Finally, they would emphasize that there is a value to this animal research on animal health, as many of these medications that have been approved for human use have also been used for the treatment of animal diseases. It's important to recognize that although companies that are developing these drugs are continuing to use animals as a model to test the safety and effectiveness of diseases, they are using alternative methods as well. Now, an example of this is called tissue culture, where we're basically going to be growing cells outside of the body in a laboratory setting. And so we can grow these cells and then test the chemicals on these cells before we move on to the animal studies. Now, an example of this is illustrated on this paper from 2023 in the scientific journal Cancers, where we're looking at taking basically a tumor biopsy from an individual over here, isolating those cells so that we can grow them in a laboratory setting, and basically how we have them on a little plastic dish here, we've got these cells growing in culture. And so what we can do through modern medicine, personalized medicine, is collect these cells from a biopsy on a patient, grow these cells in the laboratory, test a variety of FDA-approved drugs on them to determine which is going to be the most effective drug for treating this tumor in this specific patient. Now, one of the limitations to the traditional tissue culture method is that you only have one or possibly a few types of cells growing in this kind of two-dimensional flat structure. And it doesn't represent what's truly occurring within the three-dimensional body. And so modifications of this tissue culture method have generated these three-dimensional, what are referred to as organ chips. And so we have an illustration of a liver chip on this slide. And so we've got basically cells that are organized in a three-dimensional structure in this artificial environment, basically kind of small, about the size of a microscope slide, kind of smaller than a deck of cards or a personal phone. And so we can have the three-dimensional structure. So we've got some liver cells here. We've got some other cells that are found within the liver there. We've got little kind of channels that flow through this, much like the blood vessels will flow through the organs of our body to deliver the drugs, deliver the chemicals that are needed. And so using this technology, we are getting better at understanding what's going to be occurring with this drug on cells within this specific organ. But again, these organ chips are only going to show what's going on within that specific organ. Another way of getting around the problem of this two-dimensional tissue culture technique is to allow these cells under specific conditions to organize into what are referred to as organoids. In this case, we've got a heart organoid over here on the left-hand portion of the slide. And so growing these cells in culture, they self-assemble, basically organize themselves into a three-dimensional structure that has a hollow cavity, much like the hollow cavity of our hearts are going to allow for the pumping of the blood. We can see that these cells are beating on their own, and so they're mimicking what would be occurring in that three-dimensional structure of a human heart. Over here on the right-hand side, for comparison, we've got a human heart, which has been removed probably for transplantation, and so you can see the beating of the heart. You can't see the hollow cavities, but inside of this are spaces, chambers of the heart, that when the heart contracts, they're going to be pumping the blood. And so these organoids then could be potentially another model system for studying the three-dimensional structure of organs in the body. The final alternative that we're going to take a look at to animal research are computer models and computer models that include artificial intelligence. Now, an example of this is an article that came out in February 2023, taking a look at recent work done by In Silico Medicine. They were looking for a treatment for idiopathic pulmonary fibrosis. This is a disease in the lungs that results in scarring. That scarring makes it difficult to breathe, and it becomes increasingly more difficult as the scarring expands. In a research study that began in February 2021, they used artificial intelligence to try to identify potential molecular targets in the body that are associated with this scarring of idiopathic pulmonary fibrosis. And so they identified a possible target, and they also generated, using its artificial intelligence, generated some small novel chemicals that they thought could have an effect at interacting with this target. Now, a short 2 years later after that started, In Silico Medicine has been granted orphan drug designation. This is a program that supports the development and evaluation of new treatments for rare diseases, and these chemicals, the chemicals that were identified through artificial intelligence, are now entering into phase 2 clinical trials. And so completed phase 1 trials, meaning that they've been tested in volunteers that don't have the disease and shown to be safe in this small number of individuals. The phase 2 clinical trials are going to test whether or not this is now safe and potentially effective in treating a small number of patients with that disease. And this brings us now to our ethical dilemma in science for this week. Is it time to stop animal testing? And so in order to answer that question, we need to consider what type of evidence should be considered by the Food and Drug Administration to justify the use of non-animal alternatives to drug testing. And with that evidence in hand, at what point should the Food and Drug Administration accept replacement of animal-based data with data generated from these non-animal alternatives? Thank you for listening to this week's Ethical Dilemma in Science. As always, please see the description below for additional information and references. I've highlighted a couple articles that I'm recommending for seeing both the arguments for and against the use of animals for drug testing. Thank you, and have a great week.